Hello, my name is Zainab Rashid, and I had the wonderful opportunity of doing research at the University of Florida at the Herbert Wertheim College of Engineering. I worked in Dr. Thomas Angelini's lab, which bridges collective cell motion with soft matter physics and biomaterials engineering. I explored a lot here while I was UF, including going to bowling with my friends, going to Kentucky Springs, and eating a lot of good food. My specific project included developing an engineered 3D model of glioblastoma invasion. Glioblastoma is an aggressive type of brain cancer that makes surgical removal difficult. My goal focuses on 3D bioprinting KR158B glioblastoma cells with a composite material, something that we call as the supercell, to develop an invasion model. The supercell mimics the actual cell, making it a good platform for this project. To execute this project, I used 3T3s as a control. I analyzed the different modes of motility for the fibroblast and KR158B glioblastoma cells. I then printed loops to see if KR158B cells contract collectively with collagen. I then embedded the cells within the supercell to see if they move together as a giant cell. To begin with, we first performed a time-lapse imaging of the cells seated in culture dishes. As we can see, the filopodia, which are the finger-like protrusions in the KR158B, are less spindly and less elongated than fibroblasts. Their movement is more sporadic. However, in the 3T3s, the movement is more controlled. Both cell types include extension of the filopodia and rapid recoil movements. Moving on, for printing the loops and boluses for the supercell, we start by lifting the cell. In this video, I am diluting the cells with DMEM media to ensure that all the cells are lifted before I begin to count them. I then mix the cells with trypan blue and count them with the hemocytometer, which is a tool used for manual cell counting underneath the microscope. After I count the cells to the desired amount, I spin them down in the centrifuge. Usually there should be a pellet of cells that form at the bottom. After, I resuspend the cells in media. For printing the loops, I mix the cells with collagen, and when I print the supercell, I mix the cells with the composite material. I then inject the solution into the syringe and prime the needle to make sure that there is liquid coming out. I then bring the syringe in the dish with microgel to the 3D printer. These microgels contain polyethylene glycol synthesized in media. We print into this liquid because it is soft yet solid enough to support cells. This gel is versatile and can be used with any type of cells, making this 3D printing platform unique. I set up the printer by initializing it so that it moves to the proper position, and then I insert my syringe. I want to make sure that the syringe is secure so I can print effectively. After the syringe is secure in place, I jog the pump and prime the needle to make sure that the collagen hasn't gelled. I then print loops or boluses depending on the experiment. I incubate the dish with the cells, and then I set them up for time-lapse imaging on the cell scope using epifluorescent or confocal microscopy. For the loops, we tested 4 different concentrations, either low or high cell concentration and low or high collagen concentration, to see how well the cells would overcome the stress yield of collagen and start to contract. In the low cells, low collagen, as we can see here, they contract pretty significantly. However, in the high collagen, high cells, they contract rapidly, but then they start to slow down towards the end. And in high cell, low collagen, it contracts but stops after time, and this may be because there might be too much surface area or the cells are too confluent. Furthermore, in the high collagen, low cells, they contract pretty consistently, but as we can see, they leave some cells behind. We confirmed if the cells being left behind were actually cells and not collagen by using fluorescent microscopy. For the supercell, we see the KR158B cells interacting with the composites. The KR cells are indicated by the red arrows. We are currently optimizing the protocol for this specific cell type to see collective behavior that may give some insight on invasion. Some next steps for this project include troubleshooting the protocol by adjusting concentration, using velocimetry vectors which show the direction of movement of the cells, and testing whether collective migration is specific to KR158B or other glioma types. Lastly, I would like to thank my PI and mentor for giving me this opportunity, UF, and the American Heart Association for funding this. I have made so many memories here and look forward to the next chapter in my life. Goodbye.